Hello, students. I hope you all had a Merry Christmas, and we're back to learn a little bit more about biology. As you might notice, it's not my apartment, but I do hope that that all of you are having a good time with family. I am enjoying time down in San Diego. Okay, so I want to do a very quick video about Punnett squares and show you how simple they can be. So let's talk about two things today: Punnett squares and test crosses. If you can understand these two things, they're going to lay a nice foundation for both Mendelian and non-Mendelian genetics, because once we understand Punnett squares and how to use them, we really can have an understanding of probability without knowing advanced mathematics. So that's nice for those of us who aren't experts in statistics. Punnett squares are going to be a nice visual way of seeing what's going on. So whenever we do a Punnett square, all we do is just draw two lines. And there's actually only 3 Punnett squares you need to know. And this is going to go really far. So there's 3 really possibilities with Punnett square. Now, with Punnett squares, for a minute, let's assume the world is Mendelian. And this is a good assumption, again, because if something is non-Mendelian, we can kind of see in what ways it's non-Mendelian and then go from there. And so in the Mendelian world, alleles, or genes on a chromosome that code for a certain trait, are either dominant, which will make capital T, or recessive. And if one individual has both the dominant and the recessive trait, we give them a special term. And hopefully you remember that. You could pause the video, see if you remember that term. Another possibility is for the individual to be have 2 of the recessive alleles. Another possibility is for them to have 2 of the dominant alleles. And those are the only possibilities. Then we start repeating. So what are these called? Okay, hopefully you paused the video and said, this one, the double capital T, we call homozygous, or being the same, dominant. This one we call homozygous recessive. And this one we call heterozygous. So hopefully you remember that. Those are definitely terms you want to remember. And sorry about that, and should be easy for you to use. Okay, so why are we putting these random letters around? Well, by organizing the alleles for a particular trait on what's called a Punnett square, we're able to see the probability that each offspring will have that certain trait. So let's use a very Mendelian example. Let's talk about pea plants. So if we had 2 pea plants, some of them had green shells, green coloration. So that's a green pea plant. And some of them had yellow. Now, most peas we see are green, so we might suspect that that's the dominant trait. In this case, that's the case. That's not always true that the dominant trait is more common in the population. There can be other factors such as natural selection or sexual selection causing the recessive trait to become more common. But for pea plants, for Mendelian example, the green trait was both dominant and more common. So because it's the dominant trait, we know that the capital T corresponds to this, to an allele with that's coding, you could say, for the green coloration, whereas the lowercase t is for the recessive trait. These t's are arbitrary, they're just for trait. You'll see different things used for different characteristics, but you can always use t's if you prefer. It's just to represent, again, the capital being dominant and lowercase being recessive. A textbook will use different examples, different textbooks will use different letters. I suggest you just use T. Very simple, T for trait. Okay, so this parent is green. but they're not a purebred green pea plant. This, as we said, was heterozygous. They have the characteristic for both the green and the yellow. So what's going to happen in the next generation? Will they all be green? Will they all be yellow? Will it be a 3-to-1 ratio like Mendel found? What will it be? Pause the video, see if you can figure out. Well, hopefully you said that it's going to be 50-50. Why is that? Well, 50% of the time, I know that's a little hard to see, sorry about that. This yellow trait will be passed on from parent 1. Parent 2 is right here. They're going to always pass on this lowercase t. So typically how we do this is we put, you can do it either way, but we put the first letter from the parent on the left. So this parent is always passing on the lowercase t to all of its offspring. There's 2 alleles. According to Mendel's laws, it's a 50-50 shot, and that's true. So in this example, it's 50-50, but both alleles are homozygous. In this case, part of the Punnett square is kind of trivial, but it's still helpful. So this one's always passing on the lowercase t. However, in 2 of the 4 offspring, we see that this is a capital T. So how did we do this? Well, for Punnett squares, you just put the letters down. So capital T goes to the 2 boxes below it, lowercase t goes to these 2 boxes below it, and then you do the same thing horizontally as well. So lowercase t goes here and here, lowercase t goes here and here. Okay, so let's see what's going on. The offspring are 2 possible types. 50% of the offspring will be like this parent. Pretty cool. 50% will be heterozygous. 50% will be homozygous dominant. These will have what we would say a green phenotype. And these would be yellow. Pretty cool. Okay, let's do another one. So in the second example, I'll finish writing yellow. Okay, for the second example, we're crossing 2 homozygous individuals. Again, they're different, so their offspring are going to be different. This is what Mendel did. And what we see is both parents will do the same exact thing. So the green trait is passed on, and this allele is passed on. And this one is passed on. So we do write the capital T down below it. And then here we write the lowercase t to the right and the lowercase t. What's happening here? All 4 of the offspring have the same exact genetics. How did that happen? Well, both were homozygous, and homozygous because their alleles are the same, even though it's 50% this allele, 50% that allele, the 50% are the same. So it doesn't matter. And so in this case, 100% of the offspring are all the same, both genetically in their genotype and phenotype. The genotype for this is all the 100% heterozygous, and the phenotype is 100% green. Okay, let's do the last example that you need to know. So the last example is, what if both parents are heterozygous? Go ahead and pause the video. Make sure you could predict. Do a correct Punnett square, just like I taught you in the first 2 examples, and say what are the genotype and phenotype ratios. Okay, let's do it together. So we'll do the capital T's first because I'm trying to do green and yellow. Okay, so this T goes down. So we do 2 capital T's. This T goes to the right. So capital T, capital T. This T goes down. Lowercase t. Lowercase t. I meant to do that in yellow. My apologies. And this T goes over. Lowercase t. Lowercase t. wow, that's surprising. We have 3 different possible offspring. That's unique. This is the Mendelian, this is another Mendelian example that he discovered, was that when he crossed, and this was the second generation, if you remember, on our video last time. The first generation looked like this, crossing the yellow and the green. The second generation, if you look, all of these get crossed again. And then we get this peculiar 2 to 1 to 1 genotype ratio. We're twice as likely to get offspring like the parents, but 2 of the offspring don't have the same genotype as the parents. 2 of the offspring are homozygous. One is homozygous dominant, the other is homozygous recessive. What's the phenotype ratio? Well, this was the 3 to 1 ratio he found that Mendel built all his laws on. And that 3 to 1 ratio is 3 traits with the dominant trait. In this case, we said capital T represented green. So 3 of them have the dominant phenotype, the green trait, and 1 of them have the recessive trait. That was a lot of information, but here's the good news. These are the 3 examples you need to know. There's not a lot of possibilities for Punnett squares. Because we only have 3 different genotypes, heterozygous, homozygous dominant, homozygous recessive, we actually don't have a lot of possible Punnett squares we need to know. So these ratios are going to keep popping up. And once you've done it a couple of times, you can do these quicker and quicker and quicker. So what to know about Punnett squares? In the Punnett squares, each parent has 2 alleles, and either one could be given. So we can figure out all 4 possible offspring. Once we do that, we could organize those results by doing something called a Punnett square, where we can visually see all possible offspring. And then from that Punnett square, we can figure out what is the genotype ratio and the phenotype ratio of the offspring. How likely are the offspring to have certain physical traits, phenotype, or genetic combinations, genotypes. Remember, phenotype, physical trait, genotype has to do with the genes. That's all for today. I hope that's a helpful picture of how to do Punnett squares. See you in the next video.